Okay, imagine a tiny mosquito just buzzing around. Seems ordinary, right?
Hmm.
But did you know that the bacteria living inside that mosquito could actually hold the key to stopping malaria?
It does sound pretty incredible when you put it like that.
Yeah, we're talking about Anopheles mosquitoes here, the kind that transmit malaria. And today, we're diving deep into this hidden world inside them, their microbiota.
Exactly. It's this whole community of microorganisms. And the research we've been looking at, thanks to our listener, really explores this from different angles. It's fascinating stuff.
It really is. So our mission today is basically to understand this Anopheles microbiota. What's in there? What does it do for the mosquito?
And crucially, how can scientists maybe use this knowledge to prevent malaria transmission?
Precisely. We've got research covering genetics, field studies, potential control strategies. It's quite a mix.
A really comprehensive look. So where should we start? This hidden ecosystem.
Let's define it first. The microbiota, that's the community of microorganisms living in or on the mosquito. Bacteria, viruses, fungi.
Right, bacteria are the most studied component, but it includes all of them. And it's not just like random passengers. These microbes often have really important roles for the mosquito itself.
Yeah, the research points to some surprising benefits for the host, like helping with nutrition, digestion,
Hmm.
even environmental tolerance.
Absolutely. And immune support, too. They can help prime the mosquito's immune system and even offer protection against parasites.
Included malaria parasites.
Including malaria parasites, yeah. These microbes live on surfaces, inside cells, in organs. They have to cope with the mosquito's immune system constantly.
Which leads to this idea of the holobiont, like the mosquito and its microbes are one unit.
Exactly. Thinking of them together as a single biological entity, it changes how you view the whole system.
Okay, so this complex community exists. How on earth do scientists figure out who's actually living inside these tiny insects? Metagenomics, I guess.
That's the key tool set, yeah. Metagenomics lets researchers study the genetic material of the entire community straight from the mosquito without needing to grow each microbe in the lab.
Which can be really difficult, I imagine.
Extremely difficult for many microbes, so this is a huge advantage.
So it's like getting a genetic blueprint of the whole microbial neighborhood at once. How does that work? What are the techniques?
Well, there are a couple of main approaches. One is shotgun sequencing, basically sequencing all the DNA present. You break it up, sequence the bits, and use computers to put it back together.
Right, the whole shebang.
Yeah, and that tells you who's there and potentially what genes they have, hinting at their functions. The other common method is sequencing specific marker genes, like the 16S RNA gene for bacteria.
That's like a barcode for bacteria.
Pretty much. It helps identify the different bacterial types and their proportions. Shotgun gives you more detail on function, though.
And I bet both methods generate just enormous amounts of data.
Oh, absolutely. You need serious computational power and bioinformatics tools to make sense of it all. It's biology meeting big data.
Fascinating. Now, one thing that really jumped out from the research was that this microbiota isn't the same everywhere. It changes geographically.
That's a really crucial point. Yeah, one study pulled together data from different continents, Africa, Asia, America, Europe, to compare the bacteria found in Anopheles.
And did they find similarities? Like a core group of bacteria always present?
They found evidence for a potential core microbiome, yeah. Certain bacteria seem to pop up consistently across the globe, but they also noted that the data, especially from the Americas, the neotropics, is still a bit limited.
So defining that universal core is still tricky.
A bit, yes. But the differences between continents were really striking. Africa had the highest number of unique bacterial genera, 82, I think it was.
Wow, 82 unique types just in Africa.
Compared to 58 in Asia, and then only 5 in America and 3 in Europe, though, again, the American data might be incomplete.
Still, that difference between Africa and Asia and then the others, it's huge.
Yeah.
What does that imply for, say, malaria control strategies?
It strongly suggests that a one-size-fits-all approach probably won't work. If you're developing a strategy based on manipulating the microbiota, you likely need to tailor it regionally.
Because the local bacteria might have co-evolved specific roles.
Precisely. They might have different metabolic capabilities linked to their specific environment and their long history with the local mosquitoes. It highlights the need for local research.
Okay, that makes a lot of sense. The research also looked at how the microbiota changes within a single mosquito's life, right? From larva to adult.
Yes, another study tracked Anopheles gambiae in Kenya through its life stages. Larva, pupa, sugar-fed adult, blood-fed adult.
What did they find? Does it change much?
Quite dramatically. First, the water where the larvae live had the highest bacterial diversity. Only some of those bacteria actually end up colonizing the mosquito gut.
So the environment seeds the initial community.
Seems like it. And then interestingly, the diversity decreases as the mosquito matures.
Yeah.
Larvae and pupa have more diverse gut bacteria than sugar-fed adults, and blood-fed adults have even less diversity.
Why would diversity go down?
Well, there's a huge shift when the mosquito transforms from pupa to adult. It basically sheds its larval gut lining during metamorphosis.
Ah, complete gut renovation.
Sort of, yeah. And the conditions in the adult gut are very different. They saw lots of photosynthetic cyanobacteria in larvae, and pupae makes sense. They live in water. But those drop off sharply in adults.
And what replaces them?
In newly emerged adults, families like Enterobacteriaceae become more common. Then, after a blood meal, which is a huge influx of nutrients but also causes stress, the community shifts again.
Adapting to the diet and the mosquito's internal state?
Exactly. Oh, and they repeatedly found a bacterium called Elizabethingia meningoseptica. Its consistent presence suggests it might have a pretty stable, important relationship with the mosquito.
A key symbiont, maybe. So that's lab mosquitoes in life stages. What about mosquitoes out in the wild? Does their microbiota look different?
Good question. Another study tackled that, looking at wild-caught Anopheles in western Thailand. They identified 24 bacterial genera across 8 different Anopheles species.
And any surprises there?
Yeah, they found 5 genera that hadn't been reported in Anopheles before, like Pheromonas and Megasphaera. It suggests we still haven't cataloged the full diversity out there in nature.
The wild holds more secrets then.
It seems so. They also noted higher bacterial diversity in one species, Anopheles minimus. And interestingly, Elizabethingia and Serratia were common finds in these wild mosquitoes too, echoing other studies.
Okay, consistency there. Was there anything linking specific bacteria to malaria infection in the wild?
Potentially. They found 5 bacterial genera, including that newly reported Megasphaera, exclusively in a single Anopheles minimus mosquito that was also infected with Plasmodium vivax.
Just one mosquito, but suggestive.
Exactly. It's just a hint. But it raises the question, could certain bacteria influence susceptibility to malaria infection in the wild? It needs more investigation for sure. But it highlights the complexity, low diversity in one mosquito, but high diversity across the whole population.
Right, okay, so we've established the microbiota is there, it's diverse, it changes. But the million-dollar question, how does it actually affect malaria transmission?
That's really the core of it all. And the research shows it can happen in basically 2 main ways, directly and indirectly.
Okay, direct effects first. How can bacteria directly stop malaria?
Some gut bacteria seem to produce molecules that inhibit or kill the Plasmodium parasites directly, sometimes without even involving the mosquito's immune system.
Like bacterial bodyguards.
Kind of.
Oh.
The papers mentioned examples like Enterobacter species delivering anti-Plasmodium compounds, a Chromobacterium strain making something that hits both dengue virus and P. falciparum.
Wow, double duty.
Yeah, and another Enterobacter from wild mosquitoes making reactive oxygen species that interfere with parasite development. Also, co-infection with Serratia marcescens seemed to reduce P. vivax development in one case.
So, quite a few examples of direct interference. What about indirect effects through the mosquito's immune system?
Right. The microbiota can sort of prime or modulate the mosquito's immune system.
Right.
A healthy, balanced microbiota might make the mosquito better at fighting off infections, including malaria.
So it keeps the immune system on its toes.
In a way, yeah. There's also a fascinating idea that the metabolic activity of the gut bacteria, specifically their redox capacity, might help maintain an environment that favors the production of anti-malarial nitrogen oxides by the mosquito.
That's complex chemistry happening in the gut. But it's not always beneficial bacteria helping out, is it? Didn't one study find a link between certain bacteria and increased infection?
You're absolutely right to bring that up. It's not always straightforward. One study did find a correlation. Higher levels of Enterobacteriaceae bacteria were associated with an increased likelihood of P. falciparum infection.
So it cuts both ways.
It really highlights the complexity. It's about the balance, the specific species present, their abundance. Disrupting the normal microbiota can actually weaken the mosquito's defenses against malaria sometimes. It's a delicate equilibrium.
Incredibly intricate.
Yeah.
But all this understanding seems to be leading towards new control strategies, like paratransgenesis. Sounds very sci-fi.
It does sound futuristic, but it's a real strategy being actively developed. Paratransgenesis is basically genetically modifying the mosquito's own symbiotic bacteria.
Modifying the bacteria. To do what?
To produce molecules that fight the malaria parasite, or maybe molecules that boost the mosquito's own immune response against the parasite. Then you reintroduce these engineered bacteria back into the mosquito population.
You turn the mosquito's internal allies into anti-malaria agents. That's clever.
It is, yeah. The goal is to make the mosquitoes less capable of transmitting the disease.
What makes a bacterium a good candidate for this kind of engineering?
Well, you need a few key things. It has to form a stable, long-term association with the mosquito. It needs to be transmitted effectively, maybe vertically to offspring or horizontally between mosquitoes, so the modification spreads.
Okay, persistence and spread.
Right. And it needs to be culturable and genetically manipulable in the lab. Crucially, the modification shouldn't harm the bacteria or the mosquito, and the anti-malaria molecule needs to be secreted properly to actually work.
Makes sense. Are there any specific bacteria that are looking promising for this?
Yes, several are being explored. Pantoa agglomerans is one that's common in Anopheles, and engineered versions have shown strong inhibition of Plasmodium in the lab. Serratia is another candidate being modified.
We mentioned Serratia earlier in wild mosquitoes, too.
Exactly. And Azea, which is found in Anopheles and Aedes mosquitoes, is also being engineered to secrete anti-Plasmodium proteins and stimulate immunity. Researchers are even looking at fungi and viruses for similar approaches.
A whole new arsenal of potential biological controls.
Yeah.
Now, there's another bacterium that kept cropping up, Wolbachia. That one's quite well known in insect biology, isn't it?
It is indeed. Wolbachia is famous for influencing insect reproduction, metabolism, immunity. It does a lot. It's common in many insects, including many mosquito types, but its natural presence in Anopheles is a bit patchy, more variable.
But it's relevant to malaria.
Very relevant. Studies have observed a negative correlation. Mosquitoes infected with Wolbachia tend to have lower Plasmodium development.
So Wolbachia infection naturally hinders malaria.
It appears so. When scientists intentionally infected Anopheles mosquitoes with Wolbachia in the lab, they saw reduced levels of P. falciparum oocysts, a key parasite stage.
And can they establish stable infections?
They've successfully done it in lab colonies of Anopheles stephensi, and those mosquitoes became resistant to P. falciparum. So using Wolbachia is another really exciting potential strategy for blocking malaria transmission.
Wow, introducing a natural blocker. Could that be more sustainable, maybe?
That's the hope. It holds huge potential. Of course, there's always the question, could the malaria parasites eventually evolve resistance to Wolbachia's effect?
Ah, the evolutionary arms race. Always a factor.
Always.
Okay, so we've seen variation geographically through life stages. What about between different species of Anopheles mosquitoes, especially those that are better or worse at transmitting malaria?
Another really important angle. Yes, one study specifically compared the gut microbiota of 4 different lab strains from the Anopheles gambio complex. These included major vectors like An arabiensis, both insecticide-susceptible and resistant strains, and also minor or non-vectors like An maris and An quadrangulatus.
So, comparing the top malaria spreaders with less effective ones, did their gut bacteria differ significantly?
They found differences in species richness, the number of different bacteria types, but not necessarily overall diversity measures. But importantly, the composition of the bacterial community, the overall mix, did differ between the major vectors, An arabiensis, and the minor non-vectors.
Interesting. And did insecticide resistance make a difference too?
It seemed to, yes. The resistant An arabiensis strain showed differences compared to the susceptible one, suggesting insecticide resistance phenotype might actually shape the microbiota.
That's a really unexpected link. Our control methods could be influencing their internal microbes, which might affect transmission.
It's a possibility that needs more digging, for sure. It shows how interconnected everything is. Despite the differences, they also identified a potential core microbiota shared across these species, including genera we've mentioned like Enterobacter, Serratia, Elizabeth Kingia.
So some common ground, but key differences too. Could those differences partly explain why some species are such good malaria vectors?
The researchers suggest it's plausible, yes. Differences in gut microbiota might well contribute to differences in vector competence. But they caution, we need more direct infection studies comparing these species and their microbiomes to really confirm that link.
It's another piece of the puzzle then. Wow, this has been a real journey into this microscopic world. So much going on inside a single mosquito. What are the main takeaways for you?
Well, first, just the sheer diversity and dynamic nature of this Anopheles microbiota. It's a complex ecosystem. Second, it's clear these microbes aren't just passive residents, they play active roles in mosquito health, immunity, and potentially transmission. Third, the variation geographically, by life stage, by species, is huge, meaning targeted approaches are likely needed.
Right.
And finally, the potential of strategies like paratransgenesis and Wolbachia is really striking. Using the mosquito's own microbial partners against the parasite, it's a genuinely innovative direction for malaria control.
It really is astounding, thinking that these tiny interactions could be leveraged against such a devastating global disease. It feels like we might be on the verge of something new, doesn't it?
Yeah.
Maybe more sustainable, targeted control methods that work with nature.
That's certainly the hope and the driving force behind a lot of this research.
It makes you wonder, what other hidden ecosystems, maybe in other disease vectors, hold similar secrets? There's this vast, unseen microbial world influencing health in ways we're only starting to grasp. If you listening want to explore this further, try searching terms like mosquito microbiome, insect symbiosis, or vector-borne disease control. There's so much more to uncover.